a real pleasure to be here, and I'm looking forward to sharing with you some recent work from our lab. So my study focuses, or our lab focuses on developmental biology and evolution of forest trees, in particular wood formation. So during my talk today, I'm going to first share with you some background about how trees undergo secondary growth and produce wood. And then I'm going to tell you about two recent studies in our lab, the first of which is focused on understanding the evolution of transcriptional networks regulating wood formation in trees. And the second project is focused on trying to understand the genes and mechanisms underlying the evolution of a novel meristem, the monocoque cambium. So secondary growth is a term that refers to the radial expansion of stems by virtue of a vascular cambium. Now, in this slide, what I'm showing you in the top here is a section through a poplar stem, and in this region is the vascular cambium. So the vascular cambium divides to give rise to two types of tissue, the first being secondary growth, here we go, wood, or secondary phloem. Now, outside of the secondary phloem, we have phloem fibers, a cortex, and epidermis. But let's look at a little higher magnification at what's happening at the cambium. So in this cross-section from a poplar stem, in this region we have the cambial zone, and we have two types of initials. One are the fusiform initials that give rise on the wood side to either fibers or water-conducting vessels. And we also have these ray initials that give rise to rays, which participate in transport radially across the stem. So this is a really nice developmental biology system because this radial organization of the tissues actually gives us a developmental history of what's happening as far as products coming from the cambium that you can actually see in tree rings, for example. But we can also use this to our advantage. For example, in this very large poplar tree, we made an incision down the bark, and when we split it, it tends to split at the fragile cambial zone, and that allows us quite easily to harvest gram or even kilogram amounts of the meristem. So this is very unusual and helps us in gene expression and biochemical assays, for example. So the evolution of secondary growth is actually quite interesting. So now extinct lycopods, horsetails, and calamites did have a vascular cambium and made various types of secondary vascular tissues. And they, of course, were responsible for producing these wonderful carboniferous forests, now extinct, unfortunately. But in modern day plants, likely a progymnosperm ancestor gave rise to, so these were trees that had wood structures very similar to modern day conifers. But my talk today is really going to focus mostly on angiosperms. And what we believe now is that the ancestral state in angiosperms was woody, so having a vascular cambium was an ancestral state. And so a few questions arise here that we're trying to address. So one is, what are the ancestral mechanisms that were regulating vascular cambia and wood formation in basal angiosperms? And secondly, secondary growth has been lost in some groups. So for example, in these water plants of the nymphales, but more conspicuously within monocots. So the first project that I'm going to tell you about is going to be aimed at trying to identify the ancestral mechanisms regulating gene transcription in wood formation in angiosperms. The work in this project, so all the primary data was gathered by Suzanne Gertela, who's here at this meeting, along with Matt and Shutong. Matt did the bioinformatics under the guidance of Vladimir Filkov in the computer science department at UC Davis. And Shutong and Mengzhou are our collaborators in Beijing. So this is a close-up now of an angiosperm phylogeny. So as I mentioned, the ancestral state is thought to be similar to the extant Ambarella, which is basically a small woody tree or bush. Now, each angiosperm order within this phylogeny that has a little tree symbol contains one or more trees. So one thing that we want to know is what are the ancestral mechanisms that allowed early angiosperms to form wood, but also how were those mechanisms diversified in all of these various orders to produce the large array of anatomy and biochemistry that we see in wood of extant species. Now, to describe this project, I need to give you a little background information about a couple of the approaches that we used. So the first is we used gene co-expression network analyses to understand these mechanisms. And an example of how this works is given by work from a group in Umeå, Sweden, shown here. They did a very laborious experiment where they took wood-forming tissues from poplar and actually took cryosections from secondary phloem progressively through the cambium and into different stages of developing xylem. What they did next was use microarrays to look at gene expression, and they grouped genes based on expression across the different tissue samples. Now, these graphs on the right show those same groups differently now. So in this graph, all of the different tissues are arrayed across the x-axis, and expression levels are shown on the y-axis. And what you can see is that they were able to identify groups of genes that showed very similar expression patterns. So for example, in this particular group, they were expressed in secondary phloem, high expressed in the cambium, and then lower expressed during wood formation. Now, one motivation for doing these sorts of studies is that often genes within these gene modules will be participating in similar biological functions. So it's a way that we can begin to take large numbers of genes and put them into functional groups based on expression. We can then describe them in various ways using annotations, but we can also integrate these modules with other data types. So it's a very nice approach, especially in organisms where we lack like tDNA insertion libraries, for example. Now, I also need to tell you about a biological approach that we used, and that is gravitropism and tension wood. So plants show wonderful gravitropisms. For example, this Arabidopsis stem was laid on its side, and it relatively rapidly was able to right itself, and it did that by cell elongation along the bottom part of the stem. Now, in the case of trees, they can no longer elongate their lignified stems, so they take another approach. So these are aspen trees growing in Utah, and they were probably pushed over by avalanche at some point, but what you can see is they were able to right their stems. Now, someone had cut one of these trees, and this is a cross-section then through the stump. And what you can see is that there was a lot more growth on what would be the upper surface of the stem. Now, it turns out that this wood, which we call tension wood, has special properties that allows it to create a tremendous contractile force to pull the tree upright. Now, I have a little movie that shows you what this looks like in action. This is about 2 weeks of growth that's been compressed into about a minute. And what you see when this poplar tree was first placed on its side, it did the same thing as the Arabidopsis stem. So the still elongating part was able to rapidly right itself. But now you can see that the lignified part of the stem is starting to rise up too, and it's doing that not through elongation, but through production of tension wood on the upper side of the stem. If we look at cross sections through one of these trees, now we can see anatomically what's happening. So this is the top part of the stem, which we term tension wood, and you can actually see where this tree was placed on its side. So it was making what we call normal wood when it was still upright. But when it transitions to tension wood, it begins making large numbers of fibers, and it makes relatively few numbers of vessel elements. And if we look at higher magnification, these fibers actually have an additional cell wall layer called the gelatinous or G layer that is lacking in normal wood or wood formed on the bottom part of the stem, which we term opposite wood. So this is especially interesting for both basic biology, but also for applications, because tension wood, because of this contractile property, is actually the primary defect for lumber and veneer. But it turns out this G layer is actually very highly enriched in cellulose and very low in lignin. So it's attractive for potential biofuels applications. Now, one thing that I don't have time to show you today is that in previous studies, we found that when we compared gene expression within the cambial zone and early wood-forming tissues of tension wood, opposite wood, and normal wood, there are large changes in gene expression. So this allows us then to use this as a perturbation of gene expression for co-expression analyses. So this is the actual experiment that I wanted to get to. So what we did in this experiment was we used gravity stimulation to perturb gene expression in a phylogenetic survey of angiosperm trees. So we basically took each species, we laid it on its side to induce tension wood and opposite wood, and then did transcript profiling of RNA preps from either tension wood and opposite wood from trees on their side, or from normal wood from upright trees. We then used a co-expression analysis to detect gene modules in each species. So this is a phylogeny of the species that we surveyed. And the strategy here was to take kind of a twofold strategy. One was to do broad taxonomic sampling. So we included Liriodendron species, so these are magnoliids, so more basal angiosperms, as well as liquid ambars or sweet gums, poplars, willows, and eucalyptus. So we did a deeper sampling in the poplars and willows, and overall, this allowed us to do comparisons at different phylogenetic distances. But another unique part of this project was that we included both North American as well as Asian relatives from the North American Asian floristic disjunction. So these are the actual species here. So for example, within the Liriodendrons, we have Liriodendron tulipifera from North America paired with the Asian counterpart, Liriodendron chinensis. Similar for liquid ambars, both in North American and Asian species. Eucalyptus, there is no Asian counterpart here included. And the same thing for Populus and Salix. So within this project, then, ultimately we can ask questions about groups of genes that might be conserved across all of these species, as well as groups of genes that might show differences between, for example, species pairs or within specific lineages. This is the approach that we used for analyzing these data from these different species. So in this cartoon here, what we're showing are two species, A and B. And we've created co-expression modules within each of the two species separately, and then we align those, and we need to be able now to identify orthologous relationships among genes within each module. And within this analysis, we give a weight to those relationships. So in this case, we have a 1-to-1 orthologous relationship between two genes in the two species, but we can also have situations where we have one gene with two or even more orthologs in the other species. So what I'm going to show you next will be analyses that will basically summarize the degree to which both co-expression and orthologous relationships exist between these networks across species. Okay, so this is an analysis for a single willow species, Salix cameloides, kind of a tough name. But what I'm showing you here, within each one of these little diamonds is a co-expressed or a co-expression module of genes. And where you see the color is simply indicating very strong co-expression relationships. Now we can do the same thing across other willow species. So here are two other willows, and you can see that we were able to identify co-expression modules within each of these species. But the trick here now is to align these. And so on the off diagonal, what we're showing is that, for example, if we look for this module in Salix babylonica, indeed we can find it. So this would be a module that's shared between these two species. And if we go down to the third species, we can find that it's also present there. So what we can do now is the same trick, but with all of the species included. And so now you're looking at everything from the more basal magnoliids through the more derived rosids, including the poplars and willows. And there are a few things that you can see. So one of the first is that, in fact, there are modules that are conserved across all of these different species. So for example, if we look at this very large module here in Liriodendron, it's found in its Asian counterpart, as well as the liquid ambars, and all the way through all of the other more derived angiosperms. So this was an interesting result in itself because, in fact, among all of these different angiosperms, there's been an amazing amount of genome rearrangement, duplication. So it was quite probable, in fact, that we would not be able to identify any signatures of commonly expressed genes across all of these different species. But we did. Otherwise, I wouldn't be here telling you about it. Another thing that you can see is that these modules, their relationships vary at different taxonomic positions. And in fact, if you just kind of fuzz your eyes a little bit, you can see that there's a lot more commonality within these different species within the same family, the Salicaceae, than there is, for example, with the Liriodendrons. So what I want to show you next is a little bit more information about some of these putative ancestral modules, as well as lineage-specific modules that were identified. So we identified a total of 7 different modules that were conserved across all of the species examined. And what you're seeing in this heat map, within each one of these different modules, we actually have the species arrayed as rows, starting from the magnoliids through to the poplars. And then along the x-axis, we actually have go terms from each of these large categories. So for example, within hormones, we would have a large number of gene ontology groups that are recognized or represented as columns. These are not actually labeled here because the labels would be very small. So a few things that you can see, first is that these are not just random assemblages of genes, but that in fact they are enriched for specific functions. and that each module is different from the other modules. So you can already see some interesting results that we didn't know or expect. So for example, module 2 here shows a very strong enrichment for genes involved in epigenetic processes. And we don't really know very much about epigenetic roles or effects in secondary growth and wood formation, but this is certainly very encouraging to begin looking more carefully at that sort of research. We can also look within different modules and ask what are the individual genes there. So for example, if we look within this module or this group of genes in module 1 that are enriched in hormone-related processes, some of the genes that we see here are involved in auxin, brassinosteroid, gibberellic acid, ethylene, jasmonic acid. These hormones have all been implicated in various aspects of secondary growth and wood formation previously. So now with this sort of data set, we can really begin to ask fundamental questions about these different classes of genes that should be relatively conserved across a very broad group of angiosperm trees. Now we also identified lineage or even species-specific modules, and I'll just show one example here. So this is actually a rosid-specific module. And when we look at this and we look at some of the genes that are enriched within the cell wall group of genes, what we find are a number of genes that have been previously identified as being very important for wood formation, including transcriptional regulators as well as genes involved in construction of the cell wall. Now, one reason that this is an interesting result is that most of the research that has gone on for wood formation has occurred within the rosids. So for example, Arabidopsis and poplars. Now what this is telling us is that some of these very well-known genes might actually be playing different roles in more basal angiosperms, and it raises the question of whether or not we'll be able to translate some of the research that has been done in rosids to some of the more basal angiosperms. Okay, so what I have mentioned so far in this first part of the talk is that we can use this comparative gene co-expression network approach to identify both ancestral as well as lineage-specific groups of genes. That these modules show enrichment for specific biological processes, and that we can now use these approaches to help guide future research and also make predictions about genes and mechanisms underlying wood biology, as well as information about how to translate information from model systems like poplar to the many tree species that lack genomic resources. And then I'm going to end with a second story about the monocot cambium. The work done in this project, so Suzanne generated the primary data for the project, and Matt did the bioinformatics. And I'm going to go through this fairly quickly because this work was recently published, and if you're interested, this is the reference. So as I mentioned previously, monocots lack a vascular cambium and do not make wood. However, there are two monocot genera, Yucca and Cordyline, that make what's termed a monocot cambium. And this is actually one of my favorite examples of this. So this is Yucca brevifolia, better known as Joshua tree, that grows in the deserts of the southwest US. It's a very important species ecologically. And as you can see, it's not, doesn't look like your standard forest tree, but it gets quite a bit of size. And it does undergo radial thickening of the stem. So in this project, we wanted to know what are the genes and mechanisms regulating this monocoque cambium and what the relationship is of those genes to genes and mechanisms regulating vascular cambia in more traditional trees with wood. So this was our approach. First, to harvest monocoque cambium tissue from these two species, then use RNA-seq to detail gene expression, and then compare those genes to expression in vascular cambia of poplar and eucalyptus. This is actually one of the plants that we use. This is a quarter-line tree, which was actually in our backyard, and this is Suzanne here acting as a scale bar. We cut this tree down, and this is a kind of a chainsaw cross-section here. This part of the stem was actually produced by the primary meristem, but all the tissue labeled here was actually made by the monocoque cambium, which is in this very outermost layer here. Similar to what I showed for the poplar tree on its side, we split this outer layer to expose the monocoque cambium, and we were able to harvest it by simple scraping of the two surfaces that were exposed. So this is a really interesting meristem. So the meristem itself and this cross-section is in this region here, and this has been stained to show lignified cells. And so towards the inside, it's making secondary vascular bundles, which are these little red groups here, as well as conjunctive tissue. And then towards the outside, it's making a secondary cortex. And these are actually the remnants of leaf bases on the very outermost part. Now, if we look at a little higher magnification now of this region, you can begin to see the cell files, which is indicating that this is a lateral meristem that's dividing to make daughter cells that give rise to these secondary vascular bundles. At higher magnification still, what you can see is a very unusual development here. So each one of these vascular bundles is formed as a kind of meristemoid, and then it's going to differentiate kind of from oldest to newest into a single functional vascular bundle. And the way these bundles are arranged is with xylem surrounding phloem, which for you anatomy buffs is an amphivassal arrangement. So it's clearly a meristem, but it's not producing wood. It's making secondary vascular bundles. So quite different from a normal tree. So what I'm going to show you now are just two examples of gene families that we examined. The first is the class 1 NOX. So this gene family encodes transcription factors that have been shown to be very important for both the function of primary meristems as well as for vascular cambia. And what I'm showing you here, this is a heat map that is showing you the expression of the 5 different major clades of this gene family in the poplars, eucalyptus, the yucca, and cordyline. So wherever there's a square with a color, that's indicating the level of expression from low to high based on the color. And what you can easily see is that all of these genes from this family are expressed in the eucalyptus and poplar cambia, which was previously known, but they're also being expressed within the monocoque cambium. The second families that we looked at are another very ancient gene family, the class 3 HD zips, as well as the Canaries. And these genes have been very well characterized in Arabidopsis in particular, where they play antagonistic roles in vascular tissue patterning and polarity. So on the left here, we're looking at the class 3 HD zips, including Revoluta, Fab, Fav, and the ATHB 8 and 15 clades. So similar to what I showed you for the class 1 NOX, these are expressed both within the poplar and eucalyptus as well as within the cordylines. but the Canades were were different and a bit of a surprise. So in eucalyptus and poplar, we know that these genes are present in the genome because we have fully sequenced genomes, but they're not expressed in the cambial zone or wood-forming tissues. And in fact, these are actually expressed within the phloem or the inner bark of the tree. Now within the cordyline and yuccas, we did see evidence of expression of Canadi orthologs. So this was quite interesting in part because we knew that in Arabidopsis, work from John Bowman and others showed many years ago that if you create a triple mutant of Canades, you begin to get changes in the anatomy of vascular bundles where you get xylem surrounding phloem, or the same amphivassal arrangement that we see in the monocot cambium-derived vascular bundles. So this certainly doesn't prove that this is responsible for this morphology, but it's very interesting and gives us something that we could potentially follow up on in trying to understand how this very interesting anatomy arises. Now, another thing that I didn't really mention yet, but is very important. So we have now examples of class 1 NOx, class 3 HD zips, Canades, as well as other genes that I didn't have time to tell you about that are expressed both within the monocot cambium as well as in vascular cambia. But all of these genes are also expressed within the primary meristem of the shoot meristem. So this actually gives us some insights which are summarized here, which is the fact that those genes are expressed in the shoot meristem likely is the reason that they're maintained through angiosperm evolution. So they're needed for primary growth, so they're not lost when secondary growth is lost, but they can be easily reactivated, both in the case of the monocot cambium as well as other angiosperms that can evolve woody growth from herbaceous ancestors. It also is telling us that the monocot cambium likely evolved in part through co-option and reactivation of genes that are also expressed in regulating the vascular cambium in more traditional trees. So with that, I believe I've already acknowledged most of the folks, including, I do need to mention Courtney Canning, who does all of the plant propagation and greenhouse work for us. These are our collaborators at the Chinese Academy of Forestry in Beijing. And I'd also like to acknowledge funding from the USDA, DOE, and the Arnold Arboretum. And I appreciate your time.